Great. Good morning, everyone. Thank you, Danny, and thank you, Andreas. It really is great to be back in Pittsburgh. We were drawn back clearly for the science in the region. Um, and as Andreas was alluding to, we're very much an evolutionary biology lab, a comparative genomics lab. And one of our favorite things to do in the lab is to sit around and think about interesting phenotypes, as you see above, that have seemed to have come about convergently, independently in different groups of species. For example, these gliding mammals are completely unrelated to each other. One is a marsupial, in fact, but you can see that they've adopted very similar anatomical structures and behaviors and things as such. We don't yet study gliding membranes, but luckily, a collaborator, Ricardo Mallarino, who's also used some of our methods, has now employed our methods to study gliding membranes in these types of mammals. So now I don't have to do it, and that's great. So as I alluded to, we are here basically driven to study evolutionary adaptations. How do these very unique structures and behaviors and biochemical adaptations come about? We care about them for many reasons. They themselves are driven by interactions with the environment, with food sources, with other species, with competition between individuals within the same species. All of these types of interactions interplay and mold life around us. That in turn can teach us, if we study the genetic underpinnings of these traits, it can teach us how we ourselves are functioning and reveal the potential for us to create new variants within our own population, perhaps, and at least to inform health and disease. And so probably the central goal of genetics, and one that I'm going to talk about today, is therefore assigning genotypes to some of these specific phenotypes that we study using these methods. Now, people have, for a very long time, been interested in assigning genotype to phenotype, right? Of course, like I mentioned, that is perhaps the central goal of genetics. As you can see, very classic methods are often used where you can take something like an adaptive coat color change and just use classical genetics and cross it to find out which changes are responsible for that change. Other popular methods look for things like bursts of adaptive evolution or positive selection, perhaps in one particular population. You can imagine maybe these are lowland populations, and this is a population of a species that lives on a mountaintop, right? And so you can scan genomes and look for regions that are exceptional in terms of divergence within them. And this is also a popular activity. What I'm mostly going to be talking about today are kind of a growing set of methods that use different timescales from those above. The ones I've mentioned so far are within populations, between individuals within a species. But with phylogenetic genotype to phenotype tracing, we're concerned more with long-term divergence between species. So you can take advantage of the larger amount of phenotypic divergence that you see over millions or sometimes hundreds of millions of years. And as you'll see in the next slide, when we're studying phylogenetic genotype to phenotype methods, one of the important features that we can bring to bear is the fact that a lot of these phenotypes came about convergently. So you can imagine on this tree here, I've highlighted a couple cartoon branches here in red. So imagine perhaps these are high altitude specialist species, and these are the low altitude cousins, right? And so what are the different phenotypes? What are the different genotypes and features that we can associate? So the goal is to assign high-level functions to genes in regulatory regions in general. Importantly, these methods that are looking at convergent traits, they rely on this convergence to provide the replication so that we have power to detect potential genotypes that are influencing these traits of interest. On a phylogenetic timescale, between any two species, you're really going to have millions and millions of changes. So finding which are those responsible is not an easy task. And having these replicate kind of experiments that nature has carried out for us, therefore becomes very important. And so the more species that have switch to your trait of interest, the more power you're expected to have. What are we trying to associate with the trait of interest? Well, really anything we can think of. Any type of gene characteristics, as I like to call them. The first most simple gene characteristic is, does this gene exist? Or does it still seem to function? Is it present or is it absent? Is it a pseudogene? Beyond that, we can look at gene family expansions and contractions. We can look at the rate of change of any particular gene in a species or branch-specific way along that tree. We could look at expression levels, the evidence of positive selection, or even very specific properties of proteins. You could imagine maybe something like the surface charge of a protein. Perhaps that's responding to a particular selective pressure in our species of interest. And the idea is that you can therefore associate these gene or protein traits with the trait of interest. And like I mentioned, this is a growing field. There's a number of different methods out there and a growing number of contributors and groups around the world who are kind of applying themselves to these phylogenetic G2P types of problems now. So basically, I think most geneticists are going to become comparative genomicists very soon, and most evolutionary biologists are already thinking along those lines. So I've highlighted one of these characteristics here because this gene characteristic will kind of be very central to the rest of the talk, and that's the rate of change. So we may all know very well that different protein encoding genes, they evolve at different rates on average. Histones being some of the most slowly evolving, for one example, and then you have lots of immune-related functions, genes that will change very rapidly, right? And we know this. And then, of course, there's a whole spectrum of rates in between. However, that's a bit too simplistic because really these proteins are constantly changing their rates of change themselves. But if you look at, in this case, 3 different species, A, B, and C, if we were to measure the rates of change, they would be different, of course. There is always variation within biology. And to the extent that sometimes certain proteins might even shift in their relative ranks to each other. And this is kind of the fodder upon which these methods are going to thrive, right? The fact that we can study these rate changes, they will essentially reflect the different evolutionary forces that are acting on proteins at any given time. For example, conservation is, of course, a very strong force acting on all proteins basically all the time. But that the degree of conservation and kind of the constraint acting also itself varies largely over time. You can move into a new environment and a particular pathway may become very, of moderate importance when it was highly important in the other environment. And that change will then lead to kind of a relaxation of constraint on proteins in that pathway. And you will see and observe, as I'll show you later, a measurable increase in the rate of change of proteins in that particular pathway. So this type of variation in all these forces, so we're seeing the action of selection, we're seeing it change and shift these rates around. So there's a lot of information in there that we can learn. So what is one practical example I can take you to begin with? I'm going to show a number of examples. The one that does a good job of kind of setting the stage is aquatic, or in this case, marine mammals. So you can imagine back in some point in time, after the end of the Cretaceous, there was some kind of generic mammal-y thing, right? That's what I've drawn here. And mammals radiated and spread out into treetops, into deserts, forests. But then a lot of mammal lineages independently crawled back into the ocean. And some of them stay there permanently, and others are just pretty much attached to it full time, even though they can still haul out. Right? And it's important to note that each of these 4 marine aquatic lineages I put up here are more closely related to other terrestrial species than they are to each other. Right? But at the same time, you can see that they have adopted traits that are similar and just very obvious to the eye. Right? They lose their body hair covering, or the hair becomes very short in the case of fur seals. They become streamlined. Their able to see underwater, the light properties are different. You know, I could go on forever. The respiratory properties are different. And the idea here in this test case is to test and see if we can identify specific genetic loci that are underlying some of these adaptive changes. And also, perhaps we can reveal some undiscovered adaptations as well. So in doing so, we begin with large phylogenies of mammal genomes, essentially. We typically use many more than this, but nowadays with the number of genomes we have, you just can't read the labels anymore, so you'll often see me kind of truncating trees just for presentation's sake. But imagine with this collection of recognizable mammals, I've kind of highlighted the aquatic lineages in blue here, you can look at the rates of change of every protein in their genomes on every specific branch of the tree and get kind of an average rate. So this is kind of the average amount of divergence you see for all species, for all genes. Once we have this tool, we can then look at individual proteins. So here's a protein involved in the sense of taste. It's called GNAC3. And you can see that this pair of aquatic species, the dolphin and the orca, seem to stand out in this alignment. Of course, the alignment's much larger than this, having more divergence than the other species here. And that's reflected in its specific phylogenetic generate it, right? You can see the larger amount of change leading to the orca and the dolphin here. And so just by eye, in this particular case, you can see there is proportionally more change in the aquatic branches than than what you would expect given kind of the genome-wide background. Now, we don't do this by eye, of course. That would be very taxing. So we start with a similar strategy. You take the gene tree, you kind of you regress out essentially, or you subtract out colloquially the average tree from the gene tree, and we're left with those residuals, so kind of the variation of that gene from the expectation, and you get these relative evolutionary rates that we use very frequently in downstream applications, right? And so now we have this normalized rate of change where 0 represents just the same amount of change you would expect to see given the average. Positive rates are faster, negative rates are decelerated or slower. And as you can see in these red species here, those are the aquatic marines. They are all faster than the expectation in this case. And of course, now that we have these quantitative values, we can do all kinds of hypothesis testing and make inferences about specific genes. In this case, GNAC3 is very unlikely to have come from the terrestrial lineage. The aquatic species are very different from the terrestrials. And we can repeat these types of comparisons and tests over all protein coding genes and over millions of potential regulatory regions as well, and generate large, now kind of ranked candidate lists for traits or for genes that seem to be associated with these traits. Now, I'm not going to go through every row of this table, probably just half of them. No, I'm just kidding. But I can summarize them on a later slide, but you can see, if you can read this, I don't know how good this projector is, there are a lot of kind of common themes in the very top genes that are more accelerated in marine aquatic mammals than in terrestrials. And what you also see is a large number of them they're accelerated due to relaxed constraint, although there are some key examples that are accelerated due to specific positive selection events going on, meaning there's been positive selection for new amino acid variants. Statistically, there is a large excess of these accelerated genes, which is nice. And as we expected, is one reason we started with this trait. It involved lots of changes throughout the entire genome. And so we were able to, basically, if we couldn't see anything here, there was no hope of seeing anything in more subtle traits. And so mission accomplished. Just to kind of briefly summarize those genes, a large number of them were involved in olfaction and the sense of taste, sense of smell and taste. This was not surprising because large groups of marine mammals like dolphins and whales have no sense of taste or smell whatsoever, right? And we knew this behaviorally for decades. But we also saw large amounts of positive selection and genes dealing with lung surfactant proteins, which might be related to the compression that they undergo when they die. But we also saw increased positive selection in marine mammals in skin and connective tissue proteins as well, which might have something to do with forming a water-tight barrier. But actually, our favorite hypothesis now is that water is full of microorganisms, and so perhaps they're just undergoing much more encountering many more pathogens than you would expect compared to terrestrial environments. Okay. So I just want to take a brief note out to point out that this type of workflow that I point out has been developed into our package, RER Converge, very much together with Maria Chiquina's group, also here at Pitt. It's been a great years-long collaboration involving all these wonderful individuals and bringing in more wonderful individuals every year. So it continues to develop. And in fact, I just love pointing this out. This study here from Berkeley made the cover of Science, and they basically relied on this package to study lifespan differences and what genes are responsible for that in these rockfish, which are very interesting. And I'll be happy to talk about rockfish afterwards. Okay. So another type of gene characteristic we like to look at is, well, a very simple one, the presence or absence of a gene clone. Okay? So many genes are lost all the time. You know, every millions of years or so, we're losing a couple genes. A lot of us are carrying olfactory receptors that are non-functional and that happen to be different from the non-functional ones that you have. Right? So there are lots of pseudogenes in every genome. But what we decided to do is sit out and score for a much larger number than the species shown here, but score genes that appear to be pseudogenes in every species and ask which of those seem to be conking out in a correlated way in a particular environment. So again, I'm going with the marine trait here. In a phylogenetic testing framework, we're able to use this likelihood models to look at the rate of gene loss specifically within aquatic lineages. So gene loss in this case is shown by an asterisk versus gene retention. When we do this in the marine environment, you get a lot of olfactory and taste receptors, which is exactly what we expected. But what I'm going to talk about briefly here is another gene that kind of jumped to the top of the list here. It's this interesting gene called PON1, or paraoxynase 1. And that has nothing to do with the sense of taste and smell, and it kind of blew our minds. So we dug more deeply into it. We looked at a much larger, we've looked at, of course, hundreds of mammal genomes now, and in every terrestrial species we've looked at, PON1 seems to be just fine. Completely perfect, no stop codons, nothing. But every time we look at a set of aquatic lineages, there's either an ancient loss for the entire clade or multiple ongoing losses or downregulation of the genes, so it's no longer expressed anymore. We look at sea otters, it's gone. The beaver, it's gone. Manatees and dugongs, it's gone. In fact, just 2 weeks ago, I looked at a brand new primate genome. And in one primate, there seems to be a stop codon. And I looked at the species, the proboscis monkey, and it happens to be the one that lives on the edge of the ocean and is known to dive underwater to escape from crocodiles and resurface like 5 minutes later. So we might be onto yet another kind of aquatic species that has lost it. But more on that to come. What does PON1 do? In typical studies of genome-wide association studies in humans, alleles of PON1 come up as being associated with heart disease and atherosclerosis, arterial disease. PON1 hangs out on HDL and LDL particles within our bloodstream. It's there right now. And the biochemists are pretty certain that it's there to kind of break down oxidized lipids. See, this red OH is bad. It makes that lipid sticky, and sticky lipids make plaques. So it's there to kind of break these down and get them out. There's another serendipitous function. This is probably a function that has been selected for over millions of years. It also happens that paraoxynase 1, where it gets its name, breaks down organophosphate compounds that we've been manufacturing since the 1940s and 50s. And that's where it gets its name. A lot of people study this enzyme for that reason, because it has something to do with pesticides and chemical warfare. So that's another interesting function that it has that it serves for us. And it's one reason that we use organophosphate compounds in agriculture. Chlorpyrifos is a very popular one. We spray fields with it. The insects don't have PON1, they die. We have PON1, we do okay. Although don't go dousing yourself with chlorpyrifos, you're not in any way immune. But you have some resistance, right? So the problem is here, there are a lot of interesting, wonderful species like manatees that live in waterways that are directly abutting fields where chlorpyrifos is being sprayed actively. And the runoff goes directly into these freshwater waterways where manatees are overwintering and hanging out for large periods of time. And that's what this map is showing, kind of the overlap between manatee protected zones, these kind of orange-red areas, and agricultural fields in green. And so that was an interesting finding that caught a bit of press. But luckily, chlorpyrifos is being very much phased out here. So their progress is being made. Now, of course, the pesticides have nothing to do with why this gene is being lost from aquatic mammals, right? So why is it? We still don't know. But we have some favorite ideas. And the one that we must always be aware of is that it could just be lost because it's no longer important once you go into the water. Very briefly, when you become a diver, you encounter large amounts of oxidative stress due to hypoxic conditions triggering inflammation. Therefore, other defenses against oxidative damage have been ramped up in diving species. So maybe PON1 is no longer necessary. But it doesn't strike me as correct, because why would you get rid of an enzyme that repairs oxidative damage when you are encountering 10 times more oxidative damage than terrestrial species? So it doesn't seem to make sense to me. It's extremely consistently lost in aquatic mammals and seems to be lost very quickly as a species becomes like a dedicated diver. So we're working off a hypothesis that it seems to be lost due to some kind of adaptive advantage for diving. In fact, when we look at different parameters that are related to diving ability, for example, myoglobin concentration in muscle, or this kind of calculated aerobic dive limit that we gathered and calculated ourselves. Basically, the more diving adapted you are, the higher the chances are that you have PON1 has been a pseudogene in your genome for a long time, right? Now, we've since actually found that a lot of these pinnipeds up here that do not have obvious genetic lesions have actually downregulated PON1's expression, right? So it also seems to be non-functional in that context. So there's more that needs to be developed here. Okay, so I'll take you through a couple more phenotypes here, if we're doing okay for time, of course. One other phenotype that we have been working on a lot is vision. And this all began by studying a set of 4 species that have become dedicated subterranean mammals. So kind of similar to going back into the water, these guys just went underground and basically never came out. So as you can see, they've also adopted kind of body plans that are similar to each other that are probably good for tunneling and hiding underground. Also, you can see their eyes are very reduced. When we look at a proteome-wide average tree with the subterraneans shown here in red, it looks as such. If we look at this one protein that's expressed exclusively in the lens, you can see there are very long branches here for the subterranean species. And in fact, when we look at their relative evolutionary rates, they're of course very highly accelerated. And this is true kind of across the board for vision-related genes in these species. There's a very high, very strong enrichment of vision-related genes that are at the top of the list that come up. There are some other genes that I won't have time to go into that might have to do with tunneling and kind of skin and things like this. But the big story here is vision. So we decided to dig a little deeper and ask what could we learn about gene regulation and eye development as well. Because one thing that we immediately noticed is that As I showed before, this lens protein is highly accelerated in subterraneans. But if you look at genes like PAX6 that are developmental transcription factors, very important for eye development, they are not accelerated in the subterranean species. But that probably just has to do with the fact that PAX6 is also important for forebrain, pancreas, and olfactory epithelium development, which these species, of course, still need. So the protein coding sequence has not been released from constraint. It continues under very strong constraint. And that's reflected in the fact that these values are all about the same. However, perhaps there are eye-specific, ocular-specific regulatory regions that reside around PAX6 that might show some kind of signature of relaxed constraint. So we undertook this study where we looked around PAX6 and scored about 150 conserved non-coding elements. We then, at the beginning, identified 3 that were shown in the literature to be important for PAX6's expression in early eye development, and then we scored them with RER Converge, and it came that exactly those 3 were the highest scoring out of the lot, right? And so we knew that there was some signal here that we could perhaps apply to other gene regions or maybe across the genome to identify regulatory regions, because that's much more the Wild West, right? We know of what a lot of proteins are doing, not all of them, but when it comes to regulatory elements, there's so much unknown that it's almost a bit scary. So we and other groups have been coming up with new methods for studying regulatory elements throughout the genome. I won't have time to go through all the different methodologies out there, but methods are getting better. We use ones just like I've described here, looking at relative rates across branches. There are other methods like phyloconverge that are using likelihood models. There are other models being developed in part by Andreas Phoenix group, looking at machine learning models predicting, enhancerness or qualities associated with enhancers across species. In this one particular, in some of these implementations, looking at these blind mammals, again, we're able to see that there is an enrichment of ones that score high near eye and ocular related tissues, but not necessarily in non-ocular tissues, which is a good thing. So this is what we expect. But at the same time, it's not quite, these methods aren't quite yet at the ability that you can just start going down the top of the list. The enrichment, I would like to see be stronger before we do too many experiments with them. However, we are already doing some experiments with them here in embryonic zebrafish. Here you can see the little eye, and we can take some of the top scoring regions and label them with a fluorescent protein here, shown in this kind of mauve. I don't know what color it is. It's pink. And when we use this particular putative enhancer to drive the fluorescent protein, you can see it lights up in different parts of the brain, one of which is the optic tectum, which is, of course, where visual processes are dealt with in zebrafish. So perhaps this has something to do with the acceleration we see in blind mammals. We're also extending this study into the clinic. We've been working at Children's Hospital here with Dr. Ken Nischel and his group. And the idea is that he sees children with congenital eye disease and abnormalities every day. And they send some of those children's DNA off to look at the typical diagnostic panels, which are almost entirely protein coding regions. And only about half of the time are they able to assign a particular mutation as being causal for that child's abnormality, right? And so one hypothesis we're working on is, well, either there are new genes that we aren't appreciating out there that are involved in ocular development, but also couldn't a lot of these mutations also be at regulatory regions, right? And those are almost completely ignored on these clinical panels. So the ones that he can't find, he sends to us, and we sequence the patient and the parents with whole genome sequencing, and then we delve through the data looking for mutations. We've only completed 2 so far, but in both we've identified pretty hot smoking gun candidates for the causal variants in their diseases. Both were in lens-specific proteins, which we went in blindly to. Sorry about that. We didn't know what was wrong with the kids. It's designed in that way. It could have been a retinal abnormality as well, but it turns out we identified these genes and they said, wow, that's amazing because they had an absent lens and the other had cataracts. Right? So we think we're onto something. In fact, we just sent out yesterday 8 more patients and their parents to continue this. Okay, another fun phenotype that we touched on recently through a study that Amanda undertook is looking at hairlessness. This is kind of a fun trait within mammals because, I mean, it's in some ways the defining trait of mammals. You know, when you take like high school biology, right? But a lot of us mammals tend to lose a lot of that hair. You know, we have hair, but most of it's gone. Let's be fair, right? And some have none at all. So if we study this trait, you'll get exactly what you would expect, right? The keratins, all these things, collagens, hair-related proteins are at the very top of the list, very much like the subterranean mammals in eyesight, right? Also within non-coding elements, they were very much enriched near keratin-associated proteins and keratins themselves compared to kind of background expectations, okay? So there's some power there to identify regulatory regions. Here, I know this is difficult to interpret, but all of these, this is a genome track. These are different protein coding genes. And here's this really big hotspot we found of conserved non-coding elements that were accelerated in the hairless species specifically. And it didn't make any sense. There's no gene here. The closest gene is a CAM kinase gene, which is of course neurological. That never sat well with us. But then we started looking at other tracks and staring a little harder, and there's this little microRNA here, actually. So microRNA 205, right? And it turns out that if you ablate microRNA 205, it has an effect on the progenitors and stem cells in hair follicles, right? So this is really pretty fun. So bam, there it was, staring us in the face the whole time. The microRNA is what caused this. We've then gone on to look for a similar pattern throughout the genome and identify, depending on where you draw thresholds, let's say about 5 other microRNAs that have clusters of hairless species accelerated regions nearby, right? And now we're working with a collaborator at Princeton, Ricardo Mallarino. This summer, they're going to be knocking out those microRNAs or doing something to them in mice, and we'll see what happens. Okay. And actually, this study led to a really fun achievement for the lab. We were featured by Stephen Colbert. He gave us about 15 seconds on his show, but it was really pretty awesome. He pointed out that their interpretation of his writer's interpretation of our study, and then made a crack about how Emmanuel Macron has a hairy chest or something. And that was it. Okay, so if we want to summarize across a lot of these convergent phenotypes, we can see different generalizations we can make, right? There's a lot of cases here of discarded features and relaxed constraint on specific elements, be it a protein coding gene itself or regulatory regions nearby. You can see that across the board. There is also evidence that there have been kind of new functions selected for, especially in this aquatic lineages, in skin and lung surfactant proteins, perhaps due to pathogen pressures. In high altitude species, I didn't talk about that today, but we saw also adaptive evolution going on perhaps in these hemoglobins and nitric oxide signaling, which has to do with vascular signaling as well. And in one unique study that I also didn't show in detail today, we studied long lifespan in mammals and saw that a lot of kind of cancer prevention related pathways were under increased constraint in long-lived and large-bodied mammals. And interestingly enough, that rockfish study I alluded to found some of these same general functions to be under increased constraint in the long-lived rockfish. So that might be kind of a general characteristic across that's required for long lifespan invertebrates. Now, I've alluded to throughout the talk already about certain challenges that are still in front of us, right? Because we continue developing these. The good thing that's on our side is we have more and more genomes, which gives us more and more power and more traits, right? But our methodology needs to keep up. Like I mentioned last slide, loss of function traits seem to be very easy to identify, right? Once you lose something, constraint is lost, its rate of change continues, and so it sticks out like a sore thumb, so those are easier to detect. Also, when different species lose the same structure, all of the loci involved in that all uniformly become under relaxed constraint. Right? However, new traits are going to be much more difficult, and we're going to need even more sensitive methods to pick up on that, because often species will come to new traits in different ways, right? And in that case, if things aren't convergent at all, we kind of lose our power to detect them. That's one reason. Also, regulatory regions are a challenge that us and other groups are working on very rigorously these days. They're different because they don't necessarily behave like protein coding genes. The transcription factor sites turn over constantly. So something might appear to have changed, but it really didn't, right? A different compensatory transcription factor binding site popped up nearby. And because they have such low information content, these motifs, it happens all the time. And that makes these regulatory regions not as amenable to study by alignments. So for one particular solution to that, tomorrow there'll be a great keynote from Andreas to give you some clues as to how the field is progressing with that. Okay. So great, I'm doing great for time. So I want to switch gears a second here and forget about traits. Okay, forget about phenotypes. Rates change no matter what, and we know that they're changing due to changing selective pressures on these functions, right? But actually, we can do another trick with these same relative evolutionary rates. We can look and see not how a particular genetic element correlates with a particular phenotype or trait. We can look and see how these elements correlate with each other in general. Why would we do this? Well, I'll show you a couple examples to lay this out. Here are two different protein coding genes, in this case out of the yeast. And cis3 and TFC3 have nothing to do with each other functionally, and we know this because the axes have different colors. That's how we know this. And as you can see, when we plot the relative evolutionary rates of these two genes, one versus the other, there's really no relationship there notable. Right? However, if we look at two genes that have the same color, so they participate together in the complement cascade in mammals, you can see that their rates of evolution correlate pretty nicely with each other across a large number of different species. And so you end up with an R value of 0.8 in this case, R squared up 0.64. And so actually in this case, we don't even need to know what these genes are to know that they probably have something to do with each other functionally. Right? We can infer functional relationships between genes even in the absence of any knowledge about what they're doing whatsoever. And in the absence of any knowledge as to what is causing these rates to change at all, right? So I'm glad I brought some batteries. I'm not going to have time to do this. Let's just keep going. Okay, so we can essentially just measure genome by genome wide, all pairs of genes within any critters we want, and kind of measure how well correlated they are with each other. And we've done this many times in different taxonomic groups now. One kind of illustration of what this could look like is these two proteins back here were involved in the complement cascade. Here they are again, C16 and CFI. They correlate well, but if you look at all the other proteins also involved in the complement cascade, there's some pretty nice high positive values here. In fact, most of them are positive. If you look at the mean correlation across this entire pathway, it's about 0.3, which is well outside what you would expect just if you were to instead choose random genes out of the genome. Right? So this is a very nice strong signal that's genome-wide. In fact, if you look at all protein complexes and pathways, overwhelmingly they show this type of signature, right? So that means perhaps we could use this to infer new proteins. Like what if there was an additional protein that correlated well with a good number of these complement cascade proteins? Well, the obvious new hypothesis would be that it also participates in the same function as these. So we have done this a number of times. In this one particular one I'm just showing here with Cambarasa, working with Adam Kwarczykowski's group, let's just say they had these genes that they knew produced a certain phenotype having to do with cell adhesion and actin dynamics. If you see red, it means that it's producing, a mutant of that gene produces the phenotype of interest. We gave them this list of genes that correlated well with these genes. And as you can see, there's a lot of red here. About half of them scored positively in their phenotypic screen, right? Whereas if you were to just choose genes randomly out of the genome, like this small set, none of them scored positively. Ultimately, I would like to have a larger set of random genes, but you're asking people to do, this is actual work, right? I just put something in the computer, right? They had to go like do months of work, so that's all I got. I'll take it. So we've done this many times and in a bunch of different groups, and continue doing this and developing this kind of evolutionary rate covariation type of method that I've been showing. And it was also kind of written up in this perspective a couple years ago. So what we see is that this evolutionary rate covariation method, or ERC, is kind of a nice complement to put in our toolkit out there. There's lots of other ways of forming hypotheses about what genes are doing, co-expression and yeast to hybrid, 100 million things out there. And this could be yet another complementary piece of information to make networks functional networks. And we continue developing this method as well. Recently, in the face of having many, many more genomes to deal with, we had to rewrite the code base, essentially done mostly by Maria and Martin here. And so now we can handle hundreds or thousands of species, whereas we really couldn't before. And it also had the nice effect of increasing the predictive power, essentially, of these data sets. A lot of the studies I showed you before were on very small numbers of species, between like 18 and 60, right? Now we're dealing with data sets that are in the hundreds, and that just goes and increases power. So this is great. In fact, here is a yeast data set on 343 yeast species. This is the very top of the list. So if you were to look at all scores, that we're looking at the tail out here. The first thing we notice is that a lot of these are physically interacting. This pair as well, physically interacting. This is nice. The very top scoring pair was not physically interacting or didn't have a direct annotation to be known to functionally work with each other, which frustrated us at first. But then we looked at another network, and actually they're just connected. They're just one step away from each other in the stream database, essentially. So they do have a similar kind of actin cytoskeletal role. And that's actually a nice thing to point out here, is that this signature is really not driven by physical interaction. Okay? It's driven by changing selective pressures acting on these genes, much in the kind of way I was showing in the first half of the talk, talking about eyesight going away or whatever trait you're looking at. It just happens that a lot of the high scores also are physically interacting, because physical interactions are also a co-functional sign, right? So the signature doesn't come from physical interaction. However, many of them are. In fact, we just put a paper out explaining that, because it was a common misconception in the field. So now a student in the lab, Jordan Little, is endeavoring to use ERC, kind of look at functional interactions across different organelles, right? This is kind of a hot topic these days in cell biology. Just to demonstrate how it could be done, there are organelles, there's this little oxidative organelle called the peroxisome, which is there to break down lipids largely. And of course, we all know the powerhouse of the cell is the mitochondria. Compounds coming out of the peroxisome will travel into the mitochondria and also kind of in a cooperative way produce ATP. When we look at ERC values, they are very high and positive within any of these given, little processes I've highlighted here. In fact, their averages are shown here. So beta fatty acid oxidation itself has a very high, significantly high value. But interestingly, even off diagonal, we see high values between these processes. So the TCA cycle has very high values with these other pathways that are entirely performed in the peroxisome. So these kind of cross-organellar relationships even show up. And so it's her hope to use the same type of inference framework to then find new relationships between protein coding genes. So she's begun constructing networks. This is, for example, a network between all annotated protein complexes in yeast, or at least the ones that have some signal. And things cluster into these little, into these functional groups by and large. For example, we take a look at this highlighted peroxisome maintenance. All these complexes have something to do with peroxisomal functions, which is great. There are also some interesting kind of crosstalk between different pathways. For example, here we have this cluster. I'll zoom in. It is a peroxisomal ABC transporter that correlates very strongly with these mitochondrial complexes, which is similar to what we showed before, has something to do with the fact that there's passing of compounds between these organelles. Now she's begun the hard part, is coming up with specific candidates that we can get cell biologists to validate for us. So she's been looking hard at the nuclear pore complex and has found this particular protein USO1 has a very strong correlation with members of the nuclear pore complex. USO1 is very much involved in the Golgi, right? So if you look up what it does, everyone will tell you the same thing. It's involved in Golgi apparatus. It's there to kind of deal with passing of vesicles between the endoplasmic reticulum up to the Golgi. It has nothing to do with the nucleus as far as we know, right? And that's how things have all been annotated. However, we started to look into kind of obscure and old papers, and for example, in this USO1 mutant, suddenly, if you look at the nucleus, here's a nice, well-behaved nucleus. In the same assay, the nucleus becomes enlarged and kind of disorganized and such. So there are hints that perturbation of this gene actually has something to do with nuclear function as well. So it's something that we are working to advance with some more testing. So if I have to sum up all of this kind of whirlwind of things I've shown you today, it's the fact that we are very much involved in creating methods to assign higher level functions to all these molecules that we've cataloged, essentially. Genomics has done a great job of discovering protein coding genes and potential regulatory elements and such. And we need, now what we need are more tools to kind of hook all these elements. We're done with the cataloging, maybe not done, but largely. And it's time to start connecting them to higher level phenotypes, right? So once I've shown you ways we can look at lifespan and eyesight and dieting and hair and blood feeding and such. So that's our hope, is to help in this kind of endeavor that all other functional biologists and genomicists are doing to try to connect things to specific phenotypes. Great. And so last, I'd just like to thank all the people who contributed to these studies, current lab members who had lots to do with all these different phenotypes, past lab members, and lots of our wonderful collaborators in funding. And thank you all for being here, and I hope everyone stays around for the evolution session afterwards. Thank you all.
All right, Nathan, thanks so much for that visionary talk. All right, so we have a couple minutes for questions. So if you have some, please come up to the microphone here.
I always have a question. And I'm going to yell. Is it?
Go for it.
I'm waiting to see if it picks up. Yes. related. One of them is, would this work in any branch of the tree of life? And the other one is, at least the first half of what you described, you're greatly aided by already having all this functional information. Is that, oh.
Yeah, yeah, and phenotypes
In
as well.
areas where
Yeah.
you know much less about, there's much less about functional annotation at this point. Could that actually help with functional annotation? In obscure fungi or,
Yeah, that's precisely our hope. So we have a new undergrad in the lab, Nico, who's actually working on getting this ERC, this evolutionary signature, working in bacteria for the first time. And so within Mycobacterium species, and our inkling is that it's going to be much more powerful than in the vertebrates, actually. When we do the ERC studies, the correlations with known functional information are much stronger in the yeasts than they are in the vertebrates. And Drosophila is like in between. Like, so
population size.
probably, yeah,
Yeah.
to the intensity of selection and the actual kind of the difference, the large number of different environments that these poor little single-celled microbes have to tolerate versus, us cushy vertebrates who are mostly, not under so much stress. That's me speculating, but basically the measurement is that the correlations are much stronger as you go kind of to simpler and simpler life forms.
You could test that. In Enterobacteria, they always know what temperature it's going to be, for example.
Sure,
As
yeah.
opposed to, but is the muddiness of the species tree, the horizontal gene transfer, going to?
That is my biggest concern, and the reason that we are only now moving into bacteria. In fact, we chose Mycobacterium because they have less horizontal gene transfer, actually.
It's always aphids.
There's always aphids, yeah. Yeah. Thanks. Thank you.
I was just wondering if you can talk a little bit more. Okay, yes. Great, thank you. I was just wondering if you can talk a little bit more about the mutation that you have found in the regulatory region, whether they are single site substitutions and whether those substitutions are in the transcription factor binding motifs, or whether there have been expansion of tandem repeats, and also whether you have looked at the possible role played by transport elements in those regulatory regions, either, in the promoter region or in the intronic region, how that affect gene expression.
Okay. Because of the echo, I missed the very beginning of your question. You were asking about regulatory regions in general?
Right, regulatory regions. So can you talk a little bit more specific
Sure.
In terms of what kind of mutation you have found in the regulatory
By
region?
and large, these are single nucleotide changes that we're able to, at least with the RER converge type of method, because it relies on an alignment, that's what we're able to score. Typical nucleotide substitution models, as you all know, don't include insertions and deletions, unfortunately. Obviously, in those types of regions, they're going to lead to stronger phenotypes, right? So that has been another frustration of ours with alignment-based methods on regulatory regions. And another reason that we want to move into different approaches to looking at regulatory regions.
So are those single site mutations mostly happening in the transcription factor binding motifs, or it's across the promoter region?
We look at everything, although one method another alignment-free method we're developing now is looking specifically only at motifs that we can measure in some way, and looking at their different properties, different characteristics, and how they may reflect. But so what I showed today was not looking specifically at transcription factor motifs, but we're looking at ones that do focus only on those.
Okay, all right, thank you.
Very much in development.
Thank you.
Thanks.
Maybe because of the echo, maybe this is.
I think, oh wait, I was told to turn this down if the echo's bad.
It's picking us up.
There, I turned down the, okay, should be good now.
I think you're already starting to answer the question I was going for. In scenarios where you don't have the alignment, in say repeat expansions or even potentially novel protein coding genes that have arisen in particular lineages, where do you see your approaches going? Like, how can you tackle those types of regions without without the alignment?
With repeat expansions themselves, I guess.
Yeah, anywhere where you have something novel arising in one lineage and therefore there's no alignment with other lineages.
Yeah, yeah. I think there's a lot that can be done there, right? Looking for methods that focus more on a gene body rather than specific tiny micro elements around it. That would be able to capture such information. I feel like the best way through all this is going to be integrating lots of different types of information. Like, can we get chromatin contact information that will therefore inform what we should be paying attention to, or at least open chromatin? Yes. So, yeah.
I wanted to push back on the statement you made that we're mostly done with the cataloging. And kind of ask, well, the question I want to ask is, I'm sure we're getting the easy cases, but there are hard things we're missing. So I'm kind of wondering, if you could pick 1,000 more animals to sequence, how would you prioritize? What do we really want to see that we don't have yet in terms of the cataloging?
Okay. My apologies. Yeah, I completely agree. We're not done cataloging. Let's make that very clear. And if I could choose another group of 1,000 species, I mean, here's the funny thing I'll say is like, they just all seem to be falling in our lap constantly due to the hard work of many other people, right? We have the Vertebrate Genomes Project now that's developing so many. I think I would like to see more dense sampling. Last night we were talking about within plants, actually. There are certain advantages to studying plants as well. And so perhaps, balancing out the tree of life in that type of way. We should be focusing on some other groups. Yeah, thanks.
All right, so we have 2 minutes left. So I'm going to take the opportunity to ask the final question, which is, I noticed that in some of your examples, some of the genes that were under selective pressure or elements under selective pressure were regulatory in nature, like microRNAs that might have multiple targets, or transcription factors that might be regulating other genes. I was curious if you have examples of where that, let's say, regulation, where that selective pressure on the regulator also relates to selective pressure on the targets of the regulator. So the binding sites of the microRNA or the binding sites of the transcription factor, whether those different types of elements, whether you have examples where they've co-evolved.
That's a great question. So for most of the transcription factors, we do not see correlated changes with the trait, like the PAX6 example. There are two exceptions. There are two transcription factors that are kind of eye-specific, and we do see them accelerate in their protein coding regions. So that would be an interesting thing to look at. Although it's more likely just due to kind of relaxed constraints, so perhaps less interesting. Of the microRNAs, that was something on our long list of things to do that we haven't gone and looked at their targets themselves. So yeah, you're foreshadowing well something that we want to be doing.